mechanisms in immunology. So we know that metabolic pathways are governing the function and the development of different types of immune cells. Again, so the best known examples here are active T cells switch from oxidative phosphorylation to aerobic glycolysis. Then short-chain fatty acids from gut microbiome modulate a lot of type of immune cells in their development. The immunometabolism is also a key area for immunotherapy. So we need that to understand the tumor microenvironment. We also need the mechanistic understanding to optimize the function of immune cells, targeting their intracellular pathways. Then for CAR T cell transfer, for cell transfer therapeutics, so how to condition them, how to optimize these cells metabolically is also a key area. Unfortunately, so the whole field of immunometabolism is using very little metabolomics at this moment. It sounds, I know, ironic, but that's the reality. So among the few studies that actually use metabolomics, the level, so they are not really taking the full advantage of the power of the technology. Let me just show you one very recent example. So this paper was published about 2 months ago by a German group. It's actually a very nice paper. So the metabolomic data showed that Methylglyoxal is secreted by myeloid-derived suppressive cells, and they actually get into T cells to suppress T cell functions. So if you look at the metabolomic data here, so first of all, we got only 540 features measured in the assay. Also, the supplemental table with the full data set is shown here. So this column is their metabolite annotation. So the annotation is very sparse. Also, I suspect, so the reason they pick out this particular molecule is because there were already several papers, several prior papers indicating the role of the metabolite in suppressing T cells. So it's a very nice paper. I'm not really picking on the paper. So this is really the current state of art that immunometabolism is utilizing very little metabolomics. The reasons are quite a few. So again, metabolite identification and annotation is limited at this moment. So sensitivity is a major issue. So a lot of metabolomic labs actually require 1 million or more cells to do this kind of experiments. Is really not feasible. So my lab was able to drive this down to under 100,000 cells, but we can do a lot better here. Interpretation is very challenging also for a lot of people. So the access to expertise and skills may not be there. Part of the reason is, so metabolomics is quite often from analytical chemistry. So if you have to talk to somebody across campus in a different building, quite often you don't really know what they are doing and you don't know what can be achieved. So with all this, so the field of metabolomics actually progressed a lot in the past 10 years. So we routinely measure more than 20,000 metabolite features in a human tissue. So this figure here is just to show you the sensitivity based on instrumentation increased more than 100-fold. So that's over a period of about 5 years. You might remember that I had some overlap between metabolome and exposome in the chemical space earlier. So here is why. Stephen Rappaport plotted out the concentration range of different molecules in our blood a few years ago. So this light blue line is the concentration range of endogenous metabolites. Food ingredients is here, and this black line is pharmaceutical drugs. Then environmental pollutants are several magnitudes lower in terms of concentration. So the type of sensitivity we see here is probably cutting the concentration limit of detection here. So right now, I would say the metabolic instruments are placing us on this line here. So this means even without looking for all these different chemicals in a typical LC-MS experiment, we actually see all these types of molecules in the data. So this brings up a very interesting point of view. So if we compare this to the history of genomics, so it took genomics about 20 years to develop from a high-throughput sequencer to where we are today. So Metabolomics is probably here. So the hardware is good enough. We still don't have good assembly. So a lot of questions are wide open. The majority of the chemicals, the metabolites we measure in the data are not really matched to current databases or the genome scale metabolic models. So John-san kind of mentioned that some of the tools for system immunology, the black box, so we call this dark matter of metabolomics. But the high resolution data actually are good enough. You can actually put them in an archive. So this is really like express sequencing tags in the early days of genomics. I would see that identification and annotation will remain to be difficult in the near future. So this is important. I'm going to come back to this later. And no single platform can cover different kind of chemistry. So a lot of challenges, but this is also means there is a lot of opportunities. It's a wide open frontier right now. So my question here is really, should we wait for metabolomics to mature before we can reap the benefit for immunology? Of course, my answer is no. So the urgency of finding infectious diseases and cancer is, as clear as ever. So I want to emphasize that immunometabolism is really limited by the tool right now. So without a good metabolomics, the field is still like, the old story group of blind men trying to figure out, what an elephant looks like. So of course, metabolomics is already contributing a lot to detailed phenotyping for human studies, including genetic effects, microbiome products, drugs, food ingredients, environmental exposures. So the signaling rule of metabolites, again, what we know is only a tip of the iceberg. So we needed to put chemistry back to comprehensive molecular models of the immune cells. Then for system vaccinology, we needed this tool to understand the mechanisms, efficacy, how the adverse effects occur, and how the immune system actually interacts with the pharmaceutical drugs. So the last point is that if you think about Human Genome Project, so Human Genome Project did not really wait for the development technologies. It's really the application that drove development technologies. So for the next few slides, I'm going to show you a few examples of how we use metabolomics in immunological studies. This is what we call MWAS. It's actually fairly easy to do. So in this study, this is about 150 patients from malaria infection. Each column is one patient. Since we can use archived samples from a freezer, so basically as long as you locate the plasma samples, metabolomics analysis takes a few weeks, so then you get the result. Here we are looking at the association between metabolites and the parasite load in the patients. So you can see several hundred metabolites were significant. And I'm just showing some examples here. So the bilirubin products actually are related to anemia and hemolysis, which is known in malaria patients. A lot of carnitine are very significant associated with the parasite load here. So this is the same example Bali mentioned briefly during his keynote talk. I just want to point out that the blood metabolome actually mirrored the disturbance of gut microbiome after antibiotic administration very nicely. So again, the red ones are people that receive antibiotics. So you start at a time point, you can see people actually get disturbed in their metabolome after the antibiotics, and then it comes back to baseline. So one outlier here is some outlier here. So we believe that a guy just lied that he did not really take the antibiotics during the study. So the last example was really human study. So we used that more on molecular phenotyping. So then a lot of people ask, which metabolites are produced by which bacteria species in the microbiome. Again, this is a wide open field, so we know very little about it. One way to approach the question is we add the bacteria to mice, one species at a time. In this particular case, we added this PUF1 bacterium to neonatal mice. So that actually protects the mice from Listeria infection. Then the same strain, but a mutant strain in deletion 1 gene failed to confer the protection. So this addition of UF1 bacteria to mice actually showed that a lot of vitamin B related metabolites were highly increased in the mice. I'm just showing a few examples here. So that is a very possible mechanism to contribute to their protection of infection. So the last example is fairly common immunotherapy. So we use a lot of corticosteroid to suppress inflammation. In this case, it's actually very sick patients. All these are septic patients diagnosed with acute respiratory distress syndrome 7 to 1 month in the disease progression. So we are looking at metabolomic data from their bronchial lavage lipids. So we had a placebo group and we have a treatment group. Then we are looking at the metabolic profile 7 days after hospitalization. So we already knew that the treatment didn't work from this study and from many larger studies, but we didn't know why. So the interesting thing is that the steroid did perturb the metabolome quite a bit. So we see a lot more significant metabolites here. But the pathway patterns actually are very similar. So that probably explains why this did not work. So this is an ongoing project. So if we think about that, this same drug and many other drugs are used in COVID-19 patient treatment. So this is potentially very direct and important approach to understand the treatment effects. So to summarize, we can already make a lot of progress in applying metabolomics to immunology. Metabolomics itself is in explosive growth right now. So our current strategy is to start a study with global profiling and target. So this is just to cast very broad net. Then we use integrated informatics to zoom in specific hypotheses. We follow that with the target chemical and biological analysis. So again, I did not really get into the details of computational method, but metabolomics, similar to genomics, it's converting to a computational science now. So for the next phase of immunometabolomics, we have to do much better. So one major goal is to bring chemistry into systematic models for both systemic and cellular immunity. So the last point is very technical. So the thing is to have robust metabolite annotation and identification. Quite often it requires good isolation of signal for fragmentation and good reference chemicals. For many compounds, for many metabolites, this is not possible. So I would see that in the long run, metabolite identification is becoming a probabilistic problem. So it is actually critical to integrate information from other domains together with the data so that we can move forward with the field. So this is also my reasoning that immunometabolomics is not simply to put metabolomics side by side with immunology. There has to be a deep integration, deep synergy to make the science work. So I would like to thank the lab members that contributed to the data shown here today. And my deep appreciation to a lot of wonderful collaborators. A special thanks to Bali Polonjo and Dean Jones also for mentoring my career development. So part of my work is supported by the NIH Common Fund Metabolomics Consortium, for which I'm a member of the steering committee right now. The consortium is using this website, so you can find a lot of information both on education and on funding opportunities. So a shout out for our Thursday session. So we have a day-long session on computational metabolomics that is under track 6, computational mass spectrometry. So I'm co-chairing that session with Jeff Schatz. So if you are interested to learn more in metabolomics, please join us on Thursday. I think I have left enough time for questions.
Sounds good. Thank you, Shuzhao. I'll give you a boosted applause.
All right, thank you.
I'm building on my virtual skills. All right, thanks for your talk. While we are waiting for attendees to ask some questions, I will start with a question. So I was just wondering, do you do mostly these immunometabolomics assay? Do you profile the plasma? Is it possible, or is it interesting to also profile metabolites inside the cells?
We actually did a lot of intracellular studies. I did not really include much data here. Actually, that mechanism of RGD depletion as a trigger for cellular stress response was done in Bolly's lab. So that was intracellular. Metabolomics right now can be applied to almost, most type of biological tissues. We have done analysis using biopsies, sorted cells, baby diapers, sputum. So I also should be a bronchial lavage lipase. So it's really any type of sample type, as long as you can get some clean isolation of metabolites.
All right, sounds great. We have a question from the attendees. What resources do you recommend for interpreting significant metabolites? Are there structured databases of non-immunometabolomic signatures or interactions, sorry?
So that's kind of ongoing research right now. So my MWAS tool for pathway network analysis, it's widely used in the field for data interpretation for untargeted metabolomics. So actually, I'm going to be shameless on promoting myself here. So I actually have the first major book in this area. So that's data analysis and the computational method for metabolomics. It's a part of Methods in Molecular Biology series from Springer Nature. So it was published in February. So if you want to have a look at that one, it's actually a fairly comprehensive guide to computational metabolomics.
All right, wonderful, thank you.